All right. Good afternoon. For those of you who don't know me, I'm Professor Ackerman, and it's my pleasure to introduce Assistant Professor Ben Hackel, who's going to tell us about engineering novel imaging and diagnostic agents from molecular perspective. Ben is a PhD colleague of mine. We trained in Dane Wittrop's lab together at MIT. He subsequently went to work with Sam Gambier out at Stanford, who as a postdoctoral fellow in radiology. And his lab at University of Minnesota has been working on this problem for the last couple of years, where he's based out of the chemical engineering and materials science departments. Thanks, Ben.
Thank you, Margie, for the introduction, and thank you very much for the invitation. It's been a very enjoyable day of excellent conversations. So now I'm very fortunate to have the opportunity to share what we are working on and have worked on in terms of engineering these synthetic ligands, proteins in particular, for the purpose of, predominantly, the purpose of molecular imaging. So taking a protein such as this shown here and modulate it in a way such that we can generate molecular images of where locations of different biomarkers are in the body at any given time point, using mouse as a research model, but ultimately doing these imaging assays in patients. So just start with one motivation slide showing some data from cancer. The data is a couple of years old, but hasn't changed dramatically if you update it basically with the new data that's available. And it's looking at the 5-year survival that patients have as a function of when their cancer was diagnosed at stage 1, 2, 3, or 4, and the thickness of the line being the cumulative percentage of people diagnosed at an early stage. So just kind of pick one case example. So prostate cancer, what you can see is that it's very thick pretty much right off the bat, and so fortunately the grand majority of patients have their prostate cancer detected at a very early stage, stage 1 or stage 2 in a very, very high majority of the cases. And partially as a result of that, you can see that their 5-year survival is extremely high. And so that's the basically best case scenario that we have in cancer right now, is basically these early stage prostate cancers. Unfortunately, there is a subset of the patients that do not have detection until it is fully metastatic, and unfortunately, as a result, the 5-year survival, as you can see, is dramatically lower in those cases. Far more dramatic and far more problematic would be essentially all the other three shown on this graph, with one example being pancreatic cancer. And what you can see is it's very, very rare in the case in which it is discovered clinically at an early stage prior to advanced development of that disease. And in fact, the grand majority of all pancreatic cancers are detected at stage 4, in which the survival is about 5% for 5-year survival. And so this basically points out two things in my mind. And one is that we need substantially earlier detection of essentially nearly all cancers. But beyond that, we also need better treatments and a better molecular understanding to treat not just the primary disease, but the metastatic disease, because predominantly it's the metastatic disease which is driving death and metastases. The number the NCI typically points out is that 90% of cancer deaths are metastatic related, as opposed to resulting from the primary tumor. And so our attack on this problem is a molecular one. And so our aim is to identify or take other people's identification of known molecular signatures of cancer and use those to inform the treatment decision, as well as develop novel treatments as well. And so just a very, very basic schematic of what this might look like is that there might be some particular biomarker present on a tumor cell, in a particular malignant tumor cell, or a tumor cell that is going to respond to some therapy that we have or can make available through additional research, that differentiates it from healthy cells in the body, as well as through benign tumor cells, which are not necessarily going to be benefiting from any treatment. And there's a very, very long list of such things, such as the EGFR family of targets, including EGFR itself, or ERB2, and the Herceptin monoclonal antibody that targets it. c-Met is emerging as a very prevalent and important biomarker in lung cancer, as well as gastric cancer as well, and probably many more than that, but the work is ongoing. CD20 for B-cell lymphoma is another biomarker. So in short, there's a long list of these molecules that are present uniquely, or at least preferentially, on malignant tumor cells, or other disease states as well, but I'll just focus on cancer for today's discussion, that can differentiate that cell versus other cells in the body, healthy or otherwise diseased, but would not respond to therapy the same way. And so what we aim to do is use this as a tool both diagnostically and prognostically to identify patients who would benefit from different types of treatment strategies, as well as a tool potentially for early detection to identify those patients in the first case as having cancer. And the approach for that is to engineer a molecule, such as a protein shown here, that will specifically target that cell and, more importantly, that molecule present in the body. And then we can do an imaging-based assay to identify where that is located. And if we do use a quantitative modality, such as positron emission tomography, we can only locate it, but we can also quantify the relative expression of that biomarker and where it's located in the body here. And that can be done, for example, using positron emission tomography, using a positron-emitting radioisotope, such as copper-64, fluorine-18, gallium-68, etc. And so what are the benefits of a molecular imaging approach? Again, we're basically after three-dimensional, truly four-dimensional information about where are molecules of a particular type inside the body. So where is the EGF receptor located in the body at any given point in time? Where is the c-Met located within the body at any given point in time? So one advantage of that is for earlier diagnosis. And so you have anatomic approaches such as the X-ray shown here, which is able to detect this lesion in the chest of this patient. But a molecular approach, such as the fluorodeoxyglucose shown here, is a metabolic approach to molecular imaging that basically images the activity of glucose transporter and the kinase, because this is internalized into cells that are actively metabolizing glucose, and it's phosphorylated once, but then is not acted upon beyond that, and is trapped inside of that particular cell. So cells, including many tumor types that are very glucose avid, will light up on a PET scan. And so the sensitivity and selectivity might be much higher here. Now, whether PET will be used for early diagnosis or not is certainly an ongoing question. But molecular imaging in general has an improved sensitivity and selectivity for early diagnosis is certainly a valid avenue. An equally important avenue is, after the disease has already been detected, the ability to molecularly characterize that and decide should this patient be more likely to benefit from chemotherapy, from local surgical resection of the primary tumor, from some molecularly targeted systemic therapy, or et cetera. So this example is akin to what I'll be talking about, but with a different class of molecules. This is taking Herceptin, a monoclonal antibody targeted to the HER2 biomarker in breast cancer, as well as several other cancer classes, radiolabeling it with zirconium-89 isotope, which is another positron emitter, but in this case a longer-lived positron emitter, because the very long circulatory half-life of the antibody requires that these images be generated quite a long time later on. But then you can go in and image patients. This is others' work being shown here, using this exact radio tracer right here, and you can detect not only the primary tumors in these patients, but all these arrows are pointing to the metastatic disease. And so now you have information, molecular information, about the location and the relative expression of HER2 throughout these patients. And so you can identify the fact that in all three cases, there's unfortunately a very high level of metastatic disease. And so simply removing the primary tumor might still be beneficial, but it's certainly not going to be sufficient to treat these patients. And so more systemic therapy, whether it be a molecularly targeted therapy, such as Herceptin itself, or a different type of chemotherapy, are going to be necessary in these particular patients. But you also get that molecular information about the fact that, for example, these tumors are HER2 avid, high HER2 expressors, and so they would potentially respond very well to this Herceptin, and so that's a means of stratifying these patients for that therapy. At the same time, however, you might note that this patient has very high uptake in the heart, and so it's a known toxicity in a very few, about 3 to 4% of breast cancer patients, where they have cardiac toxicity associated with Herceptin, and so that would be potentially a red flag for this particular patient. But the additional assays would be required to explore that. But again, it's the molecular level of information driving the treatment decision. Another one is after the treatment decision is made, deciding what the appropriate treatments are. So you can get this information via anatomic imaging, certainly such as CT, to see that several months post-Gleevec treatment for this particular patient, you have tumor shrinkage, which is excellent. But coming back to this FDG approach, you can see that you have dramatic reduction in the glucose avidity, and research has shown that that is correlative to an excellent response to therapy, which you can see months later by the shrinkage of the tumor, but this information could be available much more readily. So you get fast and much more quantitative information about the response to treatment via these molecular-based approaches to imaging. In addition, outside of the actual clinical setting, or even perhaps in companion to the clinical setting, you can then ask questions in fully intact systems. So moving away from the Petri dish and into at least intact animal settings to ask molecular questions about the very complex disease states, whether it be for oncology or other purposes. So from a scientific perspective, molecular imaging would be very powerful. Not necessarily purely outside the clinic, though, because for drug development purposes, this is also very important. As a tool to stratify the patients for a clinical trial for your molecularly targeted therapy of interest, you are much more likely to have success when you've stratified those patients that are going to respond to your molecular treatment, as a means of pushing that molecule successfully forward for the appropriate patient population. So personalized medicine done at the level of clinical trials, so you have a much higher likelihood of success of that entity. And so the overall aim here is using molecular imaging. Again, what that is described as the localization and in most cases quantification of molecules or molecular processes inside the body. And that requires a variety of different techniques. To be molecular, you certainly need molecular recognition of that biomarker, of that molecular process. But it's insufficient to have high affinity or high specificity to your molecular biomarker of interest inside of the test tube. You need that molecule, that detection agent, to traverse throughout the body and identify where that biomarker is located. And equally importantly, you need it to traverse outside of the body or away from the other sites. And so you have the absence of signal in the cases where you have the absence of biomarkers. So you need full delivery of the molecule, and then you need anti-delivery of the molecule, except in the locations upon which it has come across a biomarker and is now localized at that site of interest. And so transport is a very, very important part here, both at the overall global level of the whole body, but also at the cellular level in terms of trafficking throughout the cell. Depending on the modality, it might matter where the actual reporter is located. So for positron emission tomography, it's not highly relevant, except in the case of perhaps non-residualizing isotopes, which might then be cleared from the cell and localized in metabolic locations as opposed to being true biomarkers. And of course, you need a detection technology to actually see where that molecule is located, whether that be positron emission tomography, optical approaches, ultrasound approaches, MRI, what have you. And then those can be applied to all the things that were mentioned. And so this field overall, our aim to improve upon this is in developing the agents themselves. And so what we want to have is a very efficient means to develop molecularly targeted agents that are as specific and as sensitive as possible. So there's really two main things here. We want this to be very, very efficient because there are numerous biomarkers. I put a very short list up there before with the EGFR family and CMET, for example, and CD20. But the list goes on and on and on, not only in oncology, but in all of the other applications where you might want to have molecular information throughout the human body or your research model. So there are very many biomarkers on this list. And as a result, we want to have available to the research community and the clinical community all the appropriate molecularly targeted agents for those biomarkers. And so we want this process from, I have a biomarker that's interesting to me, to having the actual molecular imaging agent to be very rapid. We don't want it to be a thesis every time we have a biomarker. We want it to be a month every time we have a new biomarker of interest. And so the efficiency is very, very important in this. And so we go to protein engineering as our tool to make this efficient. But equally, if not more importantly, is the need for them to truly be sensitive and specific. So optimizing that delivery is very, very important as well. So we spend a good chunk of our time looking at what sort of molecules will actually be specific in their delivery. And so we have many choices available to us. If we have, for example, a whole host of biomarkers, and our problem statement is we want ligands that bind to them specifically and with whatever the appropriate affinity and sensitivity are for our assay of interest, we have many choices. We have nature's dominant solution, the antibody molecule. And then we have a whole class of molecules that people have come up with to address this issue of ligand binding in the context of a smaller package that does not have these long plasma half-lives of an antibody, for example. We have alpha bodies, anticalins, anchoring repeats, and the list goes on and on and on. But in short, we want are molecules that are evolvable to whatever the appropriate affinity is. And for these oncology applications, for imaging, high affinity is what's needed. We need good pharmacokinetics, and we'll discuss shortly what we mean by that. Of course, we want them to be stable to work with both at the production level but also inside the human body. We need to be able to put our reporter molecule attached to this delivery agent, and so easy chemical conjugation is beneficial. And of course, if it's easy to produce, that's convenient as well. And so from the standpoint of sensitive and specific, we go here to another lab mate of Margie and I. Mike Schmidt and Greg Thurber in Dane Wittrop's lab made a very nice model of basically what happens when you inject a molecular entity into the bloodstream at a level of detail sufficient to give useful information about what sort of molecular characteristics might be beneficial, but without so much parameter space as to be lost in the uncertainty of the parameter values. And so you have injection of your imaging agent into the bloodstream. It can clear through standard physiological mechanisms. It can also extravasate out of the tissue into either healthy or tumor tissue. in the presence of biomarker, it can bind and undergo standard cellular trafficking there. And importantly, if you then put equations behind all of these arrows, you can then ask questions about what happens as I change the affinity of my molecule, what happens as I change the size of my molecule, what happens as I change the charge or the hydrophilicity of my molecule, etc. And perhaps two of the most important are going to be the binding strength, the affinity for when it's in the presence of biomarker, as well as the relative expression of the biomarker in the healthy and the tumor tissue. Is it better to have very, very unique biomarkers that aren't all that prevalent but are very unique? Or would you rather have something that's very high expressed but has some level of healthy expression as well? And thirdly, the size is very important because getting out of the vasculature into poorly vascularized, for example, small micrometastases of solid tumors is the key limitation to solid tumor targeting. And you need to have sufficient molecule to reach the tumor so that you have appropriate signal so it's sensitive, yet you need any molecule that does not encounter biomarker to be cleared so that your background signal is very, very low. You don't want a very long-lived half-life, so kind of the opposite of the standard approach to therapeutic versions. And so what you get out of this model, if you ask the question at an early time point, is instead of asking it from an area under the curve time point, is that you want the molecules to be as small as possible. So as on a log scale, as your molecular weight goes from being the antibody region out here to being kind of the mid-sized protein region here, to the 5 to smaller kilodalton molecular weight here, you can see that the tumor to background ratio being plotted on this z-axis continues to improve and improve. So the smaller your protein is, the faster it clears the background, so the background signal is very low. But importantly, it also has sufficient tumor uptake because it's able to extravasate quite rapidly from the bloodstream and diffuse to the tumor tissue. And the affinity is also important. So while it needs to be a small entity, it still needs to have relatively strong binding strength to remain bound at the site of interest and not diffuse away and be cleared from the system. And so in short, what we want are to have very small, relatively high affinity. By high affinity, low nanomolar to picomolar dissociation constants being useful. So there's some experimental support now for the benefit of small size. So this is a 10,000 molecular weight fibronectin domain that we engineered at Stanford before my time moving to Minnesota in Sam Gambier's lab, targeted towards CD20, again, a biomarker on B-cell lymphoma. And here is rituximab, the monoclonal antibody FDA approved, radiolabeled with copper-64 the exact same way. And this is a subcutaneous xenograft of the CD20 overexpressing cells in the upper left shoulder of these two mice. And what you can see is both molecules do an excellent job of targeting the CD20 positive tumor. But as I alluded to before, the small protein molecule here clears quite readily from the rest of the tissue, with slight exception in the kidney region here, whereas the antibody has a very long-lived half-life in the plasma, and so you don't get any real specificity there. Very similar data from the affibody group shown here with the comparison of an antibody and an 8,000 molecular weight affibody showing much better specificity than compared to the large molecule here. So in short, we want these molecules to be very small from a transport perspective. But that then is countered by the fact that the smaller it is, the less material we necessarily have to work with to engineer this novel binding affinity. So you look at a full antibody, and it's able to modulate the amino acid sequence here to enable to provide binding towards the relevant biomarker of interest. When we shrink down to this fibronectin domain, we can change the same amount of total surface area, but instead of it being a small fraction of our protein, it's the dramatic portion of our protein. And so intramolecular interactions are just as important, and the concern about destabilization is very, very high. And that's kind of highlighted here about the need to have the interaction energy be kind of optimized for the appropriate strength of affinity that you want. And so again, we have many, many choices, but we're now faced with the balance between needing to have good intermolecular interactions for the affinity of interest while not destroying the beneficial intramolecular interactions that we started with. And so we're left with the question then of what compound will serve as a general base case to do this with. Again, the antibody we feel is not a good solution to this problem for the size reasons explained before. At the same time, what protein is small enough to have good transport, but big enough and stable enough to be very robust? And so I will basically tell two different stories of our efforts in these two areas. One towards improving the efficiency of development. So how can we generate ligands that are in as efficient a manner as possible? That'll be using this fibronectin domain I mentioned briefly before, as well as our efforts to improve the sensitivity and specificity by working with an even smaller domain that we've identified through basically rational approach to finding the scaffold, and then a combination of rational and combinatorial to screening for these evolved ligands. And so we'll start with the story on the left first. So this is the fibronectin domain we saw in that CD20 imaging a slide ago, it comes from the full human fibronectin. So full human fibronectin is a multi-domain protein. The 10th type 3 domain located right here contains the RGD peptide motif that dominantly drives binding to integrin, several different integrin classes. But what we've done is scrubbed the amino acid sequence in this region up here such that it can be evolved to bind to new targets of interest. That work has been out and published for some time. What I'll tell today is our efforts on focusing how do we really improve the speed of finding binders from this scaffold in the context of an improved version that we made that's even more hydrophilic than the starting point, and so that it behaves much better in terms of physiological clearance. And so again, this is a very effective agent, as was shown at the CD20 imaging before, as well as some EGFR imaging that we did, both of these being Stanford work. If it'll jump ahead, then the question in protein engineering is if our aim is to have some stable scaffold, and we want this to be the same general class for all of our solutions. So as we have a new biomarker of interest, we don't have to reinvent the whole process. We always come back to the same starting point, and we're asking the question of what amino acid sequence should we put here to provide new functionality, so it will bind to its relevant biomarker and have the appropriate stability and natural clearance inside the body, etc. So the question comes to which chemistries do we put here? Which amino acid sequences do we put? Where do we place them? And that really boils down to three basic things. What location should the diversity be? Where should I actually change my starting molecule? What should I change it to, chemically speaking? I've got the 20 natural amino acids. I can go beyond that if I want to use non-natural amino acid approaches. That's possible. That's not something that we actively do. And also, what should the topology be? Should I use this fibronectin domain, or should we evolve yet another domain to achieve these aims? And that's complicated basically by the fact that the sequence to structure to function relationship is very poorly known for systems as complex as even these small proteins are. So from a purely rational design standpoint of an entire new interface, it's a very difficult problem that others are making strides on, but we still, I think, need to essentially resort to rational library design and then combinatorial high-throughput screening to find the best candidates. And so a lot of thought needs to go into what that starting library looks like to improve the probabilities of success. And so we and others have shown that you can certainly bias yourselves favorably by putting the appropriate chemistries in there. So Dev Studio at Genentech showed the benefits of tyrosine being very, very important. And we and others have put those into these non-antibody scaffolds and showed that they indeed perform much better. You also can change the local topology. So rather than picking a completely new scaffold, you can at least change the local shape of that scaffold by changing, for example, the lengths of the loops that exist on that protein. But as you'll see in the second portion, you can also work with a completely different underlying topology. But the main new information here is looking at the sites. And so if you think of the interactions, again, the natural binding protein, the antibody, it has diversity and doesn't really show up as a different color on this image here, but it has diversity in what I call the complementarity determining regions here at the tips of the antibody, and that is the dominant driving force for the binding to every new biomarker of interest inside the body. And so it changes that. And the approach is somewhat similar in these non-antibody scaffolds, where you take a region of the protein, such as the surface of this 3-helix bundle or the exposed loops on this fibronectin domain, and you essentially whitewash it and put new chemistry in. And you might use these tyrosine-biased approaches to put an appropriate chemistry in, but it's still largely a whitewash of a whole region of the protein. But if instead we actually ask the question, what do antibody repertoires actually look like? So this is a schematic looking at basically the sequence diversity as a metric of the Shannon entropy being a metric for that, the sequence diversity of the heavy chain of the protein, of the antibody, and the light chain of the antibody. And the reddish, pinkish regions are highlighting the complementarity determining regions. And what you see is it's not necessarily this whole whitewash, with the exception of this particular loop in the heavy chain of the antibody, which is essentially fully diverse throughout the human repertoire, sampling essentially all natural amino acids quite well. And then regions around that are more stabilized. But there's kind of two exceptions, right? Not all of the non-binding regions are completely conserved as the same thing. There's many different versions of antibodies that are solutions. And likewise, at all of the other loops in the antibody, they're not complete whitewashes like H3. They're actually some pretty heavy biases towards particular amino acids at particular positions. So the highly evolved solution to this diversification question is to have perhaps modest diversity even in this stable underlying core of the protein and not having complete whitewash diversity in the other portion of the protein. Yet when you look at what people are doing in the field for a lot of these different scaffold approaches, it's exactly the opposite. It's a complete conservation of the core of the protein, and it's a complete whitewash of the remainder of the protein. And so we hypothesize that if you go site-wise, even through these regions that seem to be important for intermolecular interaction, and ask site-wise questions about what would benefit, you can have a much more effective library design that's going to be much more likely to yield high-affinity stable binders in a much faster time. And so you can use a variety of tools to ask yourself which sites should be more conserved, which sites should be more diversified, and what should they be conserved diversified to. So you can use structural information to ask about the likely accessibility towards the target. You can do either experimental or computational stability to ask about which amino acids are tolerated at different positions. And you can look for homologs. If you happen to be working with a protein that has a lot of homologs available, you can go through the genomes and the proteomes to look at what amino acids are acceptable at different sites, to basically guide yourself towards maybe wanting to conserve particular positions that are conserved in nature and fairly buried and fairly important for stability while randomizing others. And so what we've done from this is used now the benefits of deep sequencing to basically ask this question experimentally. So we use those last few tools as guides towards which sites might benefit more or less from different levels of diversification. But ultimately, then, we need to ask this question in the truly complex system of the protein and the library of proteins to see which ones will work. And so we allow different sites to vary anywhere from full conservation to full diversification in different locations in between. And we put all of these different library designs simultaneously into the same test tube. So we have multiple libraries of fibronectin domains at which different highlighted sites have diversities ranging in this whole region. There's a few sites where we already have a little bit more information about, and so we put select designs there, but we won't worry about those details. In short, most of the sites in this protein have the opportunity to vary anywhere from being heavily conserved towards wild type to being heavily diversified. And as we saw from that entropy plot, some of them will benefit from either end of the spectrum. And so what we can do is simply experimentally ask which ones are more efficacious. And so we use our protein evolution toolkit to basically ask these questions. So we put all of these libraries and the different designs, briefly summarized on the last slide, into the fibronectin domain at the DNA level. And then we use Baker's yeast surface display, as our high-throughput toolkit to find which of these sequences actually have functionality. So very briefly, what this does is it takes the DNA encoding for our particular fibronectin protein of interest, diversified in whatever way that particular plasmid was diversified, and fuses the ultimate protein product to a yeast-mating protein called agglutinin 2P. As a result, the blue fusion protein docks itself on the surface of the yeast cell, and so you now have a 5-10 micron bead, a yeast cell, coated with 10 to 100,000 copies of that protein of interest on its surface, which is now a very nice linkage between the functionality of the protein and the DNA encoding for that particular functionality on the inside of this cell. And so you can use fluorescent technology. So if you take your biomarker of interest, you take c-Met receptor and you fluorescently label it, and you can then directly interrogate which sequences create proteins that actually bind to my target of interest, which ones don't, using fluorescence. You can use an assay that Margie developed to do magnetic selections via the same way. So instead of fluorescent target, you make magnetic target, and you can do very high throughput selections that way. Or you can directly take your cells of interest in a dish. You can take a petri dish full of metastatic tumor cells and a petri dish full of healthy cells, for example, and compare which proteins bind to the metastatic cells and do not bind to the other cells, for example. And ultimately, then you can pull out the DNA that encodes for those relevant proteins. You can pull out the ones that are functional. You can pull out the ones that are not functional. If necessary, you can truly do evolution. You can iterate this process numerous times to make the molecules better and better. Although for most of this initial question, we just wanted to see which libraries worked the best, and so very few cycles were needed to at least find reasonable affinity binders so that we could then do deep sequencing to find out what do the sequences look like of highly functional proteins. And so we iterated this process for about 8 different protein-based targets, some cancer biomarkers, some model proteins that we had in the lab, and then we did Illumina sequencing to generate several million DNA sequences that encoded for binders that were very high affinity as well as modest affinity, throwing away all the ones that had no functionality whatsoever. And then we could assay those by writing just a very brief bit of bioinformatics code to basically interrogate at every site throughout the fibronectin what is the prevalence of each individual amino acid in the context of binding proteins. So basically red here is a good thing. At sites where red shows up quite heavily, that means that library benefits from having a high propensity of that particular amino acid. So if we made a library that matched that distribution, it'd be much more efficacious than others. And so a few examples here. For example, this site right here, we already had from a computational approach, there's reason to believe that aspartic acid, D, would be a beneficial amino acid to have there. And so it was present at a reasonably high level in the initial library. But in the binding clones that we found, it elevated even beyond that. So what that suggests to us is that very high conservation, if not complete lockdown of aspartic acid, would be a wise thing at that site. But other sites showed a very different result. So, for example, arginine 80 right there in the protein, the initial hypotheses didn't suggest a strong reason to conserve wild type, and so we didn't, and it didn't even go up even from the low level that it had. In fact, it showed a very significant amount of many different amino acids. So in short, we learned that at this site, a heavy diversification of amino acid options are needed to give appropriate chemical diversity to bind to many different targets and different solutions to the problem. Whereas at this particular location, a heavier degree of conservation was true. And then there were sites that fell somewhere in between. But we now have detailed information for every site in our protein across binding to numerous different epitopes and numerous different targets, what the right sort of distribution looks like to be functional in the context of a fibronectin. And that has created now molecules that a library of molecules for which we barely have to do evolution, but we get very high affinity binders, usually just direct selection from this initial library using this new design. You can also look at the sites that we had reason to think were favorable of high diversity, but again, coming back to this, high diversity doesn't necessarily mean an equal level of all chemical options, but rather a distribution. And so this is just showing that the distribution that nature uses in the antibody repertoire is a pretty good utility in structures that are much, much smaller, but from a local topology, quite similar. In fact, that we have these high tyrosines and so on being seen again. So now we'll move on to the other element of it, and this is aiming for even more sensitive and more specific molecules than, for example, the fibronectin domain or other topologies might offer to us, and what ultimately yielded this new 5,000 molecular weight protein scaffold. And so, as a reminder, just a copy of the earlier slide showing the importance of the protein being very small from a transport perspective. And so, what I did was I asked an undergraduate student in the lab a couple of years back, I want to find a mini fibronectin. Please go to the protein data bank, look through the structures that were there. I think it was 89,000 or so at the time. We're now over 100K, right? Look through all of them in an efficient manner and find me a mini fibronectin. I want it to have at least 2 exposed loops. I want it to have a stable core underneath. Let's probably make it a beta sandwich to be just like this fibronectin domain that we had before. And a variety of other lists. And we collectively figured out what sort of things should be in our algorithm. And so we started with size. We can go from the nearly 100,000 at the time to a much smaller number if we simply say this needs to be small. And so what we decided on is we didn't want to be overly greedy, so I said let's not look at ultra-small things because the ability of those to robustly bind with a very high affinity might be asking a little bit much. And so we put a low cap on it just to get rid of all the numerous small peptides that are in the PDB at 40. And we said 65 was a reasonable cap. We prefer it to be much smaller than that, but we don't want the window to be too small. So we said 65, which was very, very fortunate as it turned out, as you'll see. And again, while many different topologies will ultimately work, and we're now actively looking at things that aren't mini fibronectins but are very different topologies, such as, not explicitly this guy, but other topologies as well, for starters, we wanted a mini fibronectin. So I said it needs to have a substantial amount of beta sheet contents. And so you can very easily put filters on to ask for the things that are small and the things that have at least, I think, 30%. Yeah, 30% was our cutoff for the amount of beta sheet content in the protein. And again, there's numerous ways to have a paratope. I showed this 3-helix bundle before with the surface being diversified. This is certainly a valid way to do things. But for now, we were trying to use this loop-based approach where we had at least 2 adjacent loops on the surface. We wanted at least 2 because data in fibronectin as well as other constructs have showed that that's very beneficial either from at least an additive standpoint, if not a synergistic standpoint. Fibronectin has 3, but we've shown that you don't need all 3 to have very high affinity specific binding, and since we want small size, 2 is a good compromise. I said I want 2 things that have loops. I want those loops to be adjacent and relatively large. Again, counter to the overall aim of a small protein, I want a lot of that protein to be available because you need a certain amount of surface area to provide high affinity binding to your target of interest. And so he took the protein structures from the data bank and mapped out basically where they were located. And we had a variety of ways that we went through looking at basically defining what we meant by adjacent. Simply touching was insufficient, so adjacent is not a completely proper word to put there, but we basically had a scoring metric for how we would define how likely these 2 regions would be to be additive, if not synergistic, in their interaction, and not wanting to create space in between that was fully conserved and potentially yielding, and cold spots of interaction that would be problematic from a binding perspective. So we preferred molecules that looked like this over molecules that looked like this. And of the regions that we were going to randomize and put new chemistry into, again, we wanted those to be relatively large, but to not be fooled by proteins that happen to have very large side chains to start with. We scrubbed all the protein surfaces down to serine as a trimmed baseline before we asked the size question. We also wanted them to be tolerant of mutation, because ultimately what we're going to do is take these two loops and we're going to randomize them using this antibody-type bias, diversity of high tyrosine, and so on. But they need to be able to tolerate that. So even though we're changing a reasonable fraction of the protein, they need to be able to tolerate that. We wanted to be able to do it in a fairly high throughput method. He opted to use the Eris web server that can compute delta G's of folding upon mutagenesis as kind of a compromise, but something that's very, very fast, so we can ask this question of many, many, many different mutants in the context of many, many different scaffolds, while acknowledging it's not necessarily as accurate as AMBER methods or other things such as that. But it was a good compromise, we thought at the time, and continue to think so. In addition, we preferred if it did not require disulfide bonds. If we wanted to go after future intracellular targets, it'd be nice if it was stable even in reducing environments. Or if we wanted to genetically introduce a single cysteine to give us a thiol handle for site-specific chemistry for our radioisotopes or contrast reporters, we preferred them to be cysteine-free. So we also put that in as a criterion that was necessary. And so that was our list of initial criteria. We've optimized this now to be more involved, but this was the list that we literally used to get the molecule I'll discuss now. And what it yielded as its top hit was this protein right here, which we call GP2, which stands for gene protein 2. It comes from T7 phage. I alluded to the fact that the 65 was convenient. This protein, as it exists in the protein data bank, is 64 amino acids long. These two loops, which are just about to not disappear but turn a little bit white, are not really necessary for stability of the protein, and so we've genetically chopped those off to yield this 45 amino acid protein that we have right now. It has two loops, each of 6 amino acids in the natural context on the surface. They're relatively speaking adjacent to each other, and so we can imagine additivity of binding. Overall, when you scrub these to serine residues, it yields 781 square angstroms of possible interface, and certainly we could get an additional interface from even the non-diversified portions. With the red in this image that'll pop back up in a moment is indicating the region that we're actually rationally randomizing in our library designs. And it scored quite well on destabilization, actually, in that the computational predictions for most mutants were essentially equivalent stability to the starting point, whereas the grand majority of scaffolds we looked at, as soon as you started to make this level of mutagenesis within the protein, they were substantially destabilized from that starting point. And so this was the top of the list, and so we went into the lab and produced it. So this is the amino acid sequence for the protein. We went into these two regions here and diversified them using a rational approach to what amino acids go where, and we also allowed them to go longer by 1 or 2 amino acids. That's what the dashes indicate here. So we basically took these two regions and put rationally designed 6, 7, or 8 amino acid peptides in each of the two loops, and then we put it into our directed evolution tool. So this is the rational approach to the DNA design. If we want high levels of tyrosine and aspartic acid and serine, as you see in antibodies, you can do triphosphoramidite chemistry, building DNA 3 nucleotides at a time. It was a little bit pricey for our liking at the time, and so you can actually just design a distribution of nucleotides, that for an order of magnitude less money, you can still get a very close approximation of what you want, and so that works. We also did make one rational stabilization mutation based on simply consensus modeling of this isoleucine to valine mutant. And so this we then ultimately put into this yeast display evolutionary toolkit. The library was met our quality control guidelines. It was 400 million members large. We've done quite a bit of optimization of yeast electroporation such that we now routinely get libraries of this size and actually quite a bit larger than this in many cases without having to crank through many, many different transformations in parallel. Fairly accurate DNA assembly. This is typically in the order of what we get from IDT without extensive purification, such that, you'd like it to be 100, but it's large enough to have a lot of molecules that do what you need them to do. And so put this into the evolutionary scheme, and as a starting point, we worked with convenient molecules. So we didn't jump straight into the oncology applications. We said we're going to look at goat IgG and rabbit IgG to see if we can differentiate between two very similar proteins. And we're also going to throw in lysozyme there for a completely different topology to make sure it can differentiate those as well. And then we iterated this evolutionary process as described before, using magnetic selections and flow cytometry to find binders to these targets. And we did the IgG ones first right off the bat, so that's what I'm showing right here. And so what we get are very high affinity binding to either of the two independent targets. Picomolar affinities in one case and low nanomolar in the other. They're very, very specific. They show negligible binding to anything else we have in the lab. These just being the first letter for a handful of different proteins. We tested them. This is goat IgG binding to the anti-goat IgG binder binding to its target. It does not bind to lysozyme or rabbit IgG or transferrin. It also doesn't bind to a host of other things that we tested later on, but don't have shown here. And likewise, the rabbit IgG binder binds only to it, but not to a very similar goat species there as well. That was all done on the surface of yeast, though, so it's important to take the protein off and make sure it's still effective and stable and soluble in its own, as itself as a soluble agent, and indeed it is. And so you can purify this from E. coli production quite readily with reasonable yields, and then do a binding assay and see that it retains essentially the exact same affinity that you had in the yeast context within the experimental noise. And surprising to us, even in light of the fact that the computational prediction suggested it would remain stable, I was optimistic that it would at least remain stable enough, but I really didn't think that changing at least 12 amino acids would leave it equivalently stable, but indeed it did. And in fact, many of the clones are more stable than the studying parental sequence that we have here. They don't melt up until the 70, if not 80 degree Celsius levels. And very importantly, after melting them, they're refoldable. You achieve essentially the exact same secondary structure you had before. Not shown here. After you thermally melt them and then bring them back to room temperature, they're every bit as active as they were. They bind to target just as well. And so that's very convenient from a radiochemistry standpoint. If we need to do radiochemistry at elevated temperatures to get our isotope in there, that's fine. The protein might temporarily unfold if we go at too high of a temperature, but upon cooling it back down, it's every bit as active as it was before. So that's a very convenient thing. So then we went beyond the model targets to ask if it could really be useful for molecular targeted molecular imaging. And so we started with EGF receptor, a very important biomarker in a host of different cancers. I won't even say them. I'll just leave the list up on the screen. And has FDA-approved agents both towards the kinase domains in terms of small molecule inhibitors, but also monoclonal antibodies. So from the standpoint of detection and the standpoint of patient stratification for being responsive to these molecular therapies, this would be a very useful agent to have in the clinic for many different patient populations. And so we went back to the same approach. We purchased some recombinant ectodomain of EGF receptor and fluorescently labeled it, and also made it magnetic. Did the evolutionary process and found binders that bind with very high affinity. So this is EGFR binding on the y-axis, and the amount of GP2 per cell on the x-axis, which you can normalize using this approach right here. And so what you can see is, while there's plenty of GP2 here, it doesn't show any binding in the absence. So it's not just binding to the reagents in the assay, it's literally binding to the EGF receptor. Much more importantly, it's truly binding to the EGF receptor because when you produce that GP2 protein by itself, it labels EGFR overexpressing tumor cells in culture, and you can block that activity using anti-EGFR agents. So it's truly molecularly specific and for the true EGFR, not just this recombinant version that we used as a tool to do the engineering in the first place. We then repeated that process towards an additional biomarker of interest, MET receptor, very important in lung cancer as well as gastric and hepatocellular as well. And in particular, we have a keen collaboration for pushing this forward for lung metastases, and we'll expand to those other areas in due time. So same idea, you create recombinant MET ectodomain, and you use that as the target of interest in your assay. And again, you find binders that ultimately bind appropriately. They bind to MET overexpressing cells, not to MET negative cells, such as Chinese hamster ovary cells. And they do so with appropriate affinities. We get these affinities a little bit stronger by doing additional maturation, but certainly feasible to move forward with. Excellent. So in summary, for this latter portion, we now have these GP2 molecules which seem to serve as a scaffold, as a platform to be able to find very small 5,000 molecular weight ligands targeted towards essentially anything we've offered them to date. And so we're going forward now and actually putting these into mice to see how they perform in vivo, as well as trying to do this deep sequencing to feed back into the information, feed the information back into that library design so it can be even more efficient. As I alluded to before, this is that same plot as before, but this now is fresh data for GP2. So this is deep sequencing Illumina data looking at, I think this was a smaller sliver of the Illumina. So this is hundreds of thousands of GP2 binding sequences analyzed site-wise in loop 1 and loop 2 for what amino acids are prevalent at different sites. And so we can feed this information back into our library design to have more stable starting points. And we can also do, and are doing actively, computational stabilization design to find out not only which amino acids are amenable to binding, but which amino acids are amenable to stabilizing intramolecularly the protein to start with. And beyond that, we're also trying to improve the hydrophilicity and the charge distribution on our protein, because we've shown before that greatly can impact the physiological distribution. And we have some rational design approaches using basically consensus-type design, but with a few wrinkles built in that I'm happy to discuss afterwards if people are interested, for how to modify the charge on the surface of the protein without hindering its solubility, stability, affinity. And we've done that already in the context of these 3-helix bundle alpha bodies, and we'll move forward to doing the same thing on these GP2s in the very near future. So I just want to thank all of you for your attention, as well as thanking the people who did the work, shown here in the picture and listed with the red names being highlighted for the ones who worked on the things I discussed today. So Max Kruzicki is the graduate student leading the GP2 efforts. Sumit Bhatnagar is now at University of Michigan as a graduate student. He's the initial undergraduate who initiated the GP2 work. Danny Woldring is a graduate student leading the protein engineering efforts on library design. And Hong Zhao, the postdoc, has led the efforts on the MET targeting for those. And the work was funded by the institutions shown here. So thank you. You didn't miss it because I failed to provide it, but it is. And so in that process, we not only screen for things that bind to the relevant target of interest, but we actively deplete against a select list of things that don't. And so it's an active area of discussion in the group of what should that list of molecules look like. Because you can go anywhere from doing serum, for example, so you get a nice source of a lot of different things, although it might not really contain the things that you really care about. Like you might want to have an EGFR binder that has no activity towards HER2 or HER3 or HER4. So we actively have depletions to drive specificity, but it's always a question of exactly which specificity do we want to ensure.
Is your library generic, or is there target-specific knowledge that you would use in setting up a pattern of amino acids that are analyzed?
Excellent question. We aim to have it be generic. So our hope is that we have a test tube create with this library that will bind towards, that contains members that will bind specifically towards whatever we offer it to offer it. Certainly the potential benefits towards classifying the targets into different bins and having a library for this type of target and this type of target. For now, we're taking a more generic approach. Although we're hedging on that a little bit insofar as what we're ultimately moving towards is a library of libraries. And so we'll have different topologies with the GP2 and the fibronectin. And I didn't get to it, which was wise, so I didn't go over time. We've now taken the next members of that list. So GP2 is the top of the list, but there's a whole bunch of other viable candidates below it. We've now taken the next 25 on that list while we've relaxed the constraint on topology. So now it includes helical bundles and helical surfaces and helices, but we've randomized the loops, and we've removed the topology constraints, and we've made the next 25 and put them all into the same flask, and we're going to go find out who wins. And so what I ultimately think is going to happen is there will be multiple topologies that all work. And some of them might work better towards concave or convex epitopes and things of that nature. We'll ultimately put them all in the same test tube, and it'll be our library of libraries that will go forward. But for now, at the deep sequencing level, et cetera, it's within the context of a scaffold, it's generic. Is that?
This is an academic question, but I'm just curious, what's your strategy for translation? Like, what do you patent? When do you patent it?
Yeah.
Compare, do you not compare?
That's a very relevant question in the last week or and a half. So you care tremendously because from what I've gathered and been told, if you don't do that appropriately, you can be as altruistic as you want. Unless you've got that taken care of, you're going to be really hard time helping the patients, because ultimately someone's got to pay for the clinical trial, and so you've got to do that. So after this, the GP2 is covered as far as the provisionals are taken care of and all of that, and so we're actively pursuing the appropriate means. And fortunately, the University of Minnesota has a very nice translational department built in that gives me all the input in this area science I know, but you just asked is all new to me as of like a couple months ago. So, but we have people in place to help me. So that didn't necessarily directly address the question, but the approach is to have not only the molecules of interest, but also the general technology covered so that we can move forward and partners can move forward making these molecules as diagnostics and prognostics and stratifiers for all the different applications of interest. Does that? Carl?
So it's a great talk, and I have immense appreciation for the work that went into that, and it looks like you've generated a really powerful tool as far as a resource from which you're going to be able to pull these types of targeting agents in the future. I've got a question about just thinking about now that you have this powerful tool, and you mentioned that one of your goals was to develop it so that it would be broadly useful going after this long list of biomarkers that are out there, and it's growing by the day. It seems like a lot of those are membrane proteins, and the screening strategies that you discussed here require a soluble target to be able to identify it. So I'm curious about your thoughts on how best to go about biomarkers that can't be expressed solubly, and then also maybe thinking forward into the future, the potential for intracellular markers as well. And is that something that's on your radar? Are you thinking about it? How do you plan to address those questions, or how might you?
Excellent. So let's take those in order. So first, thank you for the beginning compliments. Second, yes, they're almost all cell membrane proteins, and our take on that is, up until now, either make them in-house or call R&D systems and order the ectodomain to come in, but 50-50, maybe a little better than 50-50, that will translate beautifully. We make the protein solubly, we throw it on the cells, and it works wonderfully. And the complement of the time, no, it does. It shows either modest or no translation. And so what we now are very actively pursuing, in fact, it's basically a full project for one of the graduate students now, it's basically evolved to that for him, is to get these cell panning approaches up and working, where we can take target as it exists on the tumor cell or the cell type of interest, and pan either using yeast display, which works. The summary statement of that slide, which I cut out unfortunately, is that if you have a tumor cell expressing 1 million EGF receptors, you can take even a modest affinity binder displayed on yeast, and you can pull that up very nicely. So Eric Schuster at Wisconsin initiated that work. It works on our hands nicely until you drop that expression level. And the expression level drops to even 100K, which is still pretty high for many biomarkers. In our hands, that does not work well. In Eric's hands, it works quite well, and we'll have to have a conversation so we can get it working better. But what we're actively pursuing now is phage display towards that approach. And we think from a shear stress standpoint that it might be better. You lose all the valency, though, and so we're actively trying to work with new ideas for multivalent phage display to improve that, basically. Third part, intracellular targets. We would love to go after intracellular targets. That's a good question from a student earlier today from Margie's group. We don't have a good solution for that yet. It's as much of a delivery problem. It's a delivery problem and an anti-delivery problem, in that you need to get your ligand inside of the cell, which is difficult, but you might be able to put polyarg or TAT or some endocytic pathway and then burst the endosome, some clever way to get it in there. But that only gets you there. And if you're doing most modalities, PET or optical or otherwise, once it's there, it's on the whole time. And so if it gets there, but it sees no target of interest, it's still there, it's still emitting the signal. So either you need a smart probe, or you need the ability to get out of the cell as well when it's not there. And the delivery was hard enough. The anti-delivery is perhaps even harder. So I would love to hear any suggestions in that space. The only approach we have so far is a smart agent for photoacoustic imaging, which was led by Shai Ashkenazi in our BME department right here. He has a very nice imaging modality. That we can do protease imaging with, because there he has a particular approach where you can take a methylene blue and turn it from a very short photoacoustic lifetime to a long one after you separate it in space. But that's only useful for degradative enzymes. And so we use it for protease imaging. For biomarker imaging in general, I'm open to suggestions.
Do you consider the antigenicity of your scaffold?
Yes, but no. So we're certainly very well aware of the importance of it being non-immunogenic for most of the applications we're interested in. At the same time, while I presented it today very much from the perspective of translation, we're also very interested in this in a fundamental scientific question. What is it about a protein topology that makes it amenable to evolution or not? And so as a result, you did not see anywhere on that list of the protein data bank algorithm that it has to be human. And in fact, this is not. This is a T7 phage protein, and so that's a very relevant question. My thoughts are evolving on this, additionally, in, certainly with today's discussions, but even before that, in that my thoughts are we want it to be very small, not only for transport purposes, but the small size perhaps helps us. We've got less overall epitopes we could possibly display, and have it be very, very stable so it's less likely to get degraded, and therefore the peptides can't get displayed in the first place. Whether that's going to be sufficient or not is certainly open for debate, and so we're very actively also pursuing now the equivalent algorithm, but for human only. But regardless, we're putting new diversity in for these binding domains. So even if we start human, we're still going to have the humanization issue with the new loops. But I've had very good discussions today of some ideas on how we can start to address those things. But we have not done that to date on this new scaffold. But it's certainly on our mind.
I was wondering if post-translational modification of some of your markers could actually affect design scaffold, in terms of affinity, let's say.
Absolutely it could. And so that's another reason why moving to these cell penning approaches is of interest to us, not only for the cases of soluble protein not being folded right, but it's perhaps some of the ones that don't translate from binding soluble ectodomain to binding cells, and not because it could be because of misfolding when it's only the ectodomain you produce. It could be because post-translational modifications are not necessarily the same. I should clarify that we do usually either make or buy the ectodomains that have been made in human cells, but again, making an ectodomain and making the full intact protein are different. But where I'm especially intrigued by that is actually post-translational modifications as the biomarker. So, for example, uniquely post-translationally modified. So mucin 1, for example, and most of the mucin family actually in lung and other cancers, it has unique glycosylation in the malignant state. And so that becomes a biomarker in itself. The tricky part, and I welcome any input, is how you get your hands on that unique biomarker itself without having, or even with having to do patient biopsies as a source of the agent. It's something we're studying to pursue but don't have anything real data to share. But in short, yes, it's a very important consideration that we have to think about.
Thank you, Ben.
Thank you.